Okay, so welcome everyone to the 48th lecture of Dr. Hadi Step 1. First of all, let me ask that can you guys hear my voice? Is my voice clear? Is my voice clear? Can you guys hear my voice? Okay. Thank you so much for making it to today's lecture. And we apologize for starting the lecture 45 minutes late. It's because we had some multiple issues with a lot of problems today. First of all, we have one of the worst snowstorms in history over here in New York. And the lines are extremely down. And we apologize, but we were trying our best to get the lecture started very early. But due to the snowstorm, our lines have been very disturbed, and we got a notice from the operators that we would not be able to use the lines properly by 9:30, 9:45, and that was the reason. And I hope you guys can understand. And once again, I apologize. And but having said that, this will not affect our lecture today in the sense that we would still be able to cover what we wanted to cover in due time. That is, we were going to start with the respiratory system. Uh, that's that. And another thing, having said that, I have another I have another quite of a sad news. The sad news is one of our students got affected with the coronavirus. Um, she was not able to make to the lectures for a long period of time. I'm not sure if she made it to the lectures today, but we I'm not sure if you guys know who the student is, but that's that. So having said that, I'm not sure if the student would like to disclose the fact that she got affected with the coronavirus. I'm not going to speak her name out loud, but having said that, are you guys staying safe at home? Because the coronavirus with the second wave, things are very crazy over here. Are you guys staying safe at home and taking the necessary precautions? Okay. As a physician, I'm sure that it's my duty to tell you guys that it's about time that you guys take the necessary precautions once again, because the second wave of the coronavirus is actually pretty bad, and most of the people are getting infected left and right. Okay, so as soon as the vaccines do not get out and everything, if it's not absolutely necessary, I would advise you guys to stay at home and do all your chores at home. And that's that. Okay. Um, our love and prayers goes out to the student who got affected with the virus and her family. And having said that, um, so how many questions did you guys do from the from the reproductive system? How many questions are you guys doing? How many questions did you guys do from UWorld? Okay, everyone else? Okay, very good. 25, 40. Everyone else? How many questions are we doing from UWorld before we begin with the respiratory system? How many questions are we doing? Okay. Out of all those questions which you guys are doing from the reproductive system, how are your percentages, score percentages? Guys, can I get some answers, please? How are your scores? How are your scores in the U world before I begin today's lecture? Everyone else, how are your scores in UWorld? We have a student with 80%. That is very remarkable. Anyone else, how are your scores? Okay. Can we get some answers from the students regarding their score percentages in UWorld, please, before we begin today's lecture? From so that we know if you guys are doing well in the UWorld. How are your score percentages? Okay. Um, okay. Having said that, are we ready to begin the respiratory system today? Because it's actually pretty late, so we just want to get ahead of schedule. Are we ready to begin the respiratory system? Yes or no? Are we ready to begin the respiratory system? Okay. Okay, all right. So having said that, let's begin with the respiratory system. Okay, the first part of the respiratory system that we're going to talk about, the first part of the respiratory system that we're going to talk about is respiratory embryology. Can you guys see my screen? Is my screen visible to you guys all? Can you guys see my screen? Is the screen visible? Can I get some answers, please? Because, okay. Okay, very well. Okay, so let's begin with today's lecture. That is the respiratory system. We are going to begin with respiratory embryology. That is the first part of the respiratory system. Over here, what we can see that there is the first part that is lung development. And lung development, usually the development occurs in 5 stages. So 5 stages. For these 5 stages, we have to know the names of the stage, which structures are developing, and if we have any pathology from each and every stage. And how important is this for your USMLE step one? The importance of this is that you guys will be tested based on your knowledge, basically on the structural development from each and every stage. Okay, so what we're going to do is we're going to do what we do best. That is, we're going to take a new page and start with the first page, and we're going to review and understand the text, master the text, add mnemonics, and make this thing very easy and digestible so that we can all understand and apply this knowledge when we solve the USMLE questions. Okay, so first of all, the stages, the 5 stages, before we talk about the 5 stages, let's talk about the embryology as a whole. Can I get some fast answers from you guys? First of all, we have the zygote formation. Okay, first of all, we have fertilization and zygote formation. After zygote formation, can you guys tell me what is the next step of development, please? Fast answers, because that's what we do from the embryology, step by step. What is the next step of development after zygote formation? Okay, good. Murulent implantation. What happens after implantation? What happens after implantation? Fast answers, please. What happens after implantation? Okay, in the second week, we have the formation of the bilaminar disc, which is the epiblast and the hypoblast. Then the formation of the trilaminar disc, am I correct? Gastrulation, which happens in the third week, that is ectoderm, mesoderm, and endoderm. Okay, ectoderm, mesoderm, and endoderm. From here, we have which layer is important for the development of the lungs when we talked about the derivatives of ecto, meso, and endo. Which one is responsible for the development of the lungs? Endoderm, very good. Endoderm. Endoderm is responsible for the development of the lungs. Okay, out of the endoderm, do we have the development of the GI structures, which is the foregut, midgut, and hindgut? Yes or no? Fast answers, please. Okay, foregut, midgut, and hindgut. From the foregut, does the foregut have ventral part and ventral And then every part of the foregut, midgut, and hindgut, they have the ventral part. Then they have which part? How many parts do they have? Ventral, and then dorsal, and then ventral, dorsal. Oh, okay. So basically, from the parts of the foregut, midgut, and hindgut, we have the foregut, which has the ventral part and the dorsal part. From the ventral foregut, we have the development. We have the development of diverticulum, So from the ventral foregut, if something like this happens over here, like let's say this is the fetus, okay, over here, okay, if this is the fetus, then over here we have this sort of diverticulum. This diverticulum is known as respiratory diverticulum. This appears from the ventral foregut, Okay, can you guys please write this down in your book? It's not there. Yeah, that the respiratory diverticulum, okay, it arises from the ventral foregut. Okay. Okay. Did you guys write it down? Did you guys write it down? Okay. So next time you guys receive a question, where is the respiratory diverticulum arising from? Can I get some fast answers, please? Can I get some fast answers, please? Where is the respiratory diverticulum arising from? Can I get some fast answers? Ventral foregut. Very good. Okay. Okay, very good. So from this respiratory diverticulum, you have the distal end. So from the distal end of the respiratory diverticulum, so we have proximal and distal. We have this is the distal end. This is the proximal end. So from this distal end of the respiratory diverticulum, you have what we call is the formation of the lung bud. We have the formation of a bud known as lung bud, which will go through the 5 stages of development. And this usually begins, okay, how many words are there in lung? How many words are there in lung? Fast answers, please. How many words are there in lungs? I would need some active participation from you guys in the chat box if you want to finish this today. Very good. 4 words in lungs. So what is the week at which lung development begins? What is the week at which lung development begins? Very good. 4 weeks. Okay. 4 weeks. So lung development begins from the distal end of the respiratory diverticulum, which arises from the ventral foregut, and it begins at 4th week, week 4. It begins at 4th week. Okay, so that's that. Now let's talk about the stages of the lung development. Let's talk about the different stages of lung development. From the stages of lung development, we have 5 stages. Okay, we have 5 stages. So out of those 5 stages, okay, the mnemonic that we're going to use is every pulmonologist can see alveoli. Alveoli. Every pulmonologist can see alveoli. With this, we have the 5 stages and the letters, that is E, P, C, S, and A. Okay, so with that, the first stage, okay, we're going to have our stages over here. We're going to have the structural development over here, structures arising from each stages, and we're going to have the pathology. Okay, so this is how we're going to study this. Okay, so first of all, we have the E4 embryonic stage. E4 embryonic. Okay, E4 embryonic stage. So the first stage is the embryonic stage, and the weeks are, since it begins from the 4th, it goes from 4 to 7. Okay, it goes from 4 to 7. Okay, are we clear? Are we clear about this? Are we clear about this part of the lecture so far? Are you guys with me? Can you guys understand what I'm talking about? Okay, please keep your utmost attention to this because right after this, I will ask you guys questions. Okay, so embryonic stage is from 4 to 7 weeks. And from this embryonic stage, we have the development of basically what's developing is, first of all, we have the lung bud, which is developing from the distal end of the respiratory reticulum. We have the lung bud. Okay, from the lung bud, what we have is, from the lung bud, we have the development of the tracheus. Okay, we have the development of the trachea. From the trachea, we have the development of the bronchial buds. Okay, bronchial buds. And then stepwise, what we have is stepwise development of the lungs. That is main stem bronchi, main bronchi, then lobar bronchi, step by step, okay, lobar bronchi, then last one is tertiary bronchi. Tertiary bronchi. Okay. So lung bud, trachea, bronchial buds. And from the bronchial buds, we have the main lobe and then tertiary bronchi. So if we talk about this, then basically what's happening is trachea, okay, then right and left main, okay, then we have the lobes of the lungs. So lobar, like this. And then over here, it gets further developed into, okay, so something like this. Okay. So this is happening over here. Keep in mind, this layer has still not formed. It's just only this part. That is this part to this part. And this part to this part is the embryonic stage. Okay. Trachea, main stem bronchi. Okay. Main stem bronchi, lobar, then tertiary bronchi. Are we clear about this? Are we clear about this? Yes or no? Okay, very good. So far, so easy. Okay, next one. Next one is, the next letter is P. P for what? P for, the next letter that is P. P stands for pseudoglandular. Pseudoglandular. Okay. Okay, P stands for pseudoglandular. So we have the pseudoglandular stage. Pseudoglandular stage. From the pseudoglandular stage, you have the development. Okay, so from the structural development, you have the endodermal tubules. Okay, you have the endoderm. You have the endodermal tubules. Okay, tubules, right? And from the endodermal tubules, what you have is the formation of the terminal bronchiole. What you have is the formation of the terminal bronchiole. Okay, so from the pseudoglandular stage, you have the endodermal tubules. From the endodermal tubules, you have the formation of the terminal bronchioles. Okay. Okay. This pseudoglandular stage this week is from week 5 to week 17. Week 5 is to week 17. Okay. Are we clear about this? Are we clear about this? Yes or no? Okay. So endodermal tubules to the terminal bronchioles. That's all that's forming in the pseudoglandular stage. Okay, it's your pseudoglandular stage. Okay, the next stage is, stay with me over here. The next stage is every pulmonologist. So we are done with E, P. The next one is C. C stands for canalicular. C stands for canalicular stage. This stage, in the canalicular stage, we have the rest of the developments. If I talk about the terminal bronchioles all the way up to the alveolar ducts, what we have is we have terminal bronchioles, then we have respiratory bronchioles, and from the respiratory bronchioles, what we have is we have the alveolar duct. We have the alveolar duct. Okay, so that's that. Okay, so this is forming the canals. So the name of the canals are the canalicular states. Once you have the formation of the terminal bronchioles, from terminal bronchioles all the way to the respiratory bronchioles to the alveolar duct, that is over here, there's further division of these ones. Okay, so this was your tertiary bronchi, right? From the tertiary bronchi, you had the development of the terminal bronchioles, right? And from the terminal bronchioles, you had the respiratory bronchioles. From the respiratory bronchiole, you had the alveolar ducts. So that's that. Okay. The canalicular stage, this is 5 to 17. The canalicular stage is 16 to 25. Okay. The weeks are not that very high yield. You will not be asked on a lot of questions based on the weeks, but the names are important. Which stage is responsible for which one? Okay, so that's that. Okay, so we are done with the canalicular stage. Okay, the next stage that we want to go to is S. S stands for saccules. The word saccules, saccular. Okay, the word saccular stage. Saccules, this basically means that you have the formation of the terminal sacs, that is the alveolar duct over here, alveolar duct to the formation of the terminal sacs. Okay, the terminal sacs. Okay, these terminal sacs are right before the formation of the proper alveoli. They are known as the terminal sacs. These terminal sacs, they are separated by a septa, that is the primary septa, but that's not very important. Just try to remember that the alveoli are still not developed at the saccular stage. What we have is we have the terminal sacs, but they're not fully matured alveoli. Okay, so this is from week 26. So this ends at 25. So this stays from 26 to up to birth. So this is the saccular stage. Okay, the last stage is extremely easy to understand. The last stage is what you have is every pulmonologist can see alveoli. So the last stage is alveolar stage. The last stage is alveolar stage. Alveolar. Alveolar stage is basically week 36 up to 8 years. 8 years. So basically, even when the baby is little, lungs are still developing. So that is the alveolar stage. Okay. Over here, it's easy. That is, you have the development of the terminal sac to adult alveoli. To adult alveoli. Okay. So that's that. That's exactly what's happening over here. Okay. So basically, before we jump into this any further, and talk about the pathologies, I will give you guys 2 minutes. I will give you guys 2 minutes, and you guys need to read these things once again. Okay, read these things once again, and once I have tested that you guys have understood and reviewed and kept this in your mind, only then shall I move forward with the pathology. Okay, because if we don't master the physiology, we can't understand the pathology that is over here. So I'll give you guys 2 minutes. It's 10:14 as of right now. At exactly 10:16, I will ask you guys the questions. Okay, so 2 minutes on the clock. Let's do this. Okay, is everyone ready? Is everyone ready? Okay. Guys, are you guys ready to answer questions? We don't have a lot of time because we want to finish the respiratory system. Okay, it's not a very big system. We can finish it very quickly. Okay, very good. Let's begin. First of all, how many stages does the lung have in the development from the lung bud? Very good. What are the 5 stages? Okay, number one. What is the number one stage? What is the number one stage called? Fast answers, please. What is the number one stage? Embryonic stage. How many weeks are needed for the development of the embryonic stage? 4 to 7 weeks. Very good. Okay. What are the structures that is developed from the embryonic stage? First of all, we have the lung bud. From the lung bud, we have the next one, trachea. From the trachea, we have from the trachea, we have bronchial buds. Very good. From the bronchial buds, we have main stem bronchi. From the main stem bronchi, we have which one? Lobar. From the lobar, we have the terminals. I mean the tertiary. My apologies. Tertiary bronchi. Okay. From there, we have the tertiary bronchi. Okay, what is the next stage called? What is the next stage called? Pseudoglandular. Very good. What are the weeks of pseudoglandular? What are the weeks of the pseudoglandular? 5 to 17. Very good. 5 to 17. What are the lung developments from the pseudoglandular stage? Which one? Endodermal tubules up to the terminal bronchioles. Very good. Up to the terminal bronchioles. Okay, very good. Then the next stage is called canalicular stage. The canalicular stage, what are the weeks of the canalicular stage? 16 to 25. Very good. 16 to 25. What are the structures developing for the canalicular stage? Terminal bronchioles, then respiratory bronchioles. Very easy to understand. After respiratory bronchioles, what we have alveolar ducts. Alveolar ducts. Okay, last stage. Not the last stage, but the last stage before the adult development. This is the saccular stage. Very good. Saccular. Saccule. The word saccule stands for the formation of the sacs. That is the terminal sacs. Okay. That is week 26 till birth. Thank you so much. And over there, we have the alveolar ducts to the formations of the terminal sacs. Okay. Last stage. What is the last stage called? Alveolar stage. Okay. What are the weeks of the alveolar stage? Weeks of the alveolar stage. Passes, please. Weeks of the alveolar stage. Week 36 up to 8 years. Very good. Okay. So what are the structures developed from the alveolar stage? Terminal sacs up to the adult alveoli. Terminal sacs to the adult alveoli. Okay. Are we all clear about this? Okay. Are we all clear about this? Okay. Very good. If we are clear, then let's begin. Okay. Okay. So the next thing that I want to talk to you guys about is, okay, the next thing that I want to talk to you guys about are the pathologies and little here and there notes which are responsible for each and every stage. Okay, so the first stage is the embryonic stage. In the embryonic stage, if we have any problem, okay, so the stage and pathology. Okay, so the first stage is the embryonic stage. In the embryonic stage, if we have a problem, okay, if we have a problem with the embryonic stage, then what we will get is tracheo-esophageal fistula. Okay, we will have tracheo-esophageal fistula, meaning that the trachea, if it's not well developed, okay, then we can get what we can call is a TEF, tracheo-esophageal fistula. We have studied a lot of different types of tracheo-esophageal fistula in the gastrointestinal system with the students who were there in the GI. Have we not? Have we not studied tracheo-esophageal system? Tracheo-esophageal fistula? Okay, very good. Okay. So tracheo-esophageal fistula, basically what this is this is the esophagus, this is the trachea, that's how it's supposed to be. But over here, if there's an anomaly, you can have a fistula like here, like this. Okay, so the food, when you eat it, it can go through this way, that's that. And this is the H type of tracheo-esophageal fistula, that is that. Then we have esophageal atrasia with pure tracheo-esophageal fistula, that's another one. So we have a lot of different types of tracheo-esophageal fistula, meaning that there's a fistula, there's an abnormal connection between the trachea and the esophagus. And this can happen when you have a problem with the embryonic stage. Okay, the next stage that we talked about was pseudoglandular stage. Pseudoglandular stage. The pseudoglandular stage, basically, this means that if we have a patient who has been born at the pseudoglandular stage, that patient will that patient will unfortunately pass away. That patient will unfortunately pass away. Why will they pass away? They will pass away because at this stage, you do not even have the formation of the alveoli here, right? All you have is the development of the terminal bronchioles, and the terminal bronchioles cannot provide appropriate gaseous exchange. So the young child who will be born at the pseudoglandular stage, that is between week 5 to 17, at this stage, he or she will unfortunately pass away. Okay, so that's that. So this at this point, the respiration is incompatible with life. Okay, the child will pass away. Okay, next stage. Next stage is canalicular stage. At the canalicular stage, we have the development of a very important. We have the development of a very important cell. Okay, we have the development of the very important cell. This cell that is being developed at this stage. Okay, this cell that is being developed at this stage is known as pneumocytes. Okay, this is known as pneumocytes. So primary pneumocytes, they developed at the canalicular stage, exactly at more or less at the 20 weeks. The pneumocytes, we have type 1, type 2 pneumocytes, the functions of which we are going to study in just a little bit. But before we do that, do we understand how important the pneumocytes are for the development of the lungs? Are they important, or are they not important at all? Very good. So we have the development of the pneumocytes. The pneumocytes are developed at the canalicular stage, especially at the 28 weeks of gestation. And another highly, the reason why this baby is laughing over here, the reason why this baby is laughing is that if the baby is born at the canalicular stage, that baby can survive. Basically meaning that at the canalicular stage, since you have the formation of the alveolar ducts, okay, from the alveolar ducts and the canalicular stage, you have terminal bronchioles with the alveolar ducts, some amount of gaseous exchange is possible. And at this point, if the baby is born at 25 weeks with appropriate care at NICU, at 25 weeks, the baby can survive. So that's that. Okay. Okay. Next one is the saccular stage. From the saccular stage, we do not have anything high yield, because at the saccular stage, of course, the baby will survive because you have the formation of the terminal sacs. But since it's from week 26 to birth, even if the baby is born after week 26, the baby will survive. But we have to keep the baby in a very close, in an intensive care unit, that is NICU. So that's that. So over here, the baby will survive. Okay, at the saccular stage. Okay. Then the last one is alveolar stage. Alveolar stage. Okay. What happens at the alveolar stage is that at the alveolar stage, the young baby, since they have the formation of the adult alveoli at week 36, after week 36, okay, okay, first of all, what is the normal time for delivery? Fast answers, please. What is the normal time for delivery? What's the normal week at which delivery usually occurs? Okay, very good. So is that more than 36 weeks, yes or no? Of course. So that's more than 36 weeks. So what happens is, in more than 36 weeks, you have already the development of the adult alveoli. So the baby, what he does is the baby starts breathing. Okay, so the baby starts breathing. What does he breathe? The baby breathes in the amniotic fluid. So the baby inspires and expires. So as a result, the baby aspires and expires amniotic fluid. When the baby inspires amniotic fluid, the fluid, it goes into the lungs and it causes lung expansion. So that's why you need expansion of the lungs. So in babies with oligohydramnios, if there is insufficient fluid, then there is pulmonary hypoplasia. So in order for proper expansion of the lungs, the babies, they breathe in and they expose the amniotic fluid. And what this does is, This increases the resistance, the vascular resistance of the lungs, right? Because if the lungs are filled with fluid in little babies, then the vascular resistance, that is the blood vessels, they will have an increased resistance of going through the lungs. Okay, but so as a result, they will keep on flowing through the placenta. But at birth, what happens is at birth, the baby, when they breathe in the air, okay, keep in mind what I'm talking about. This is really, really important. At birth, when the babies, when they breathe air, they have a sudden decrease in pulmonary vascular resistance. Since they have a sudden decrease in pulmonary vascular resistance, now the blood from the right ventricle can go all the way to the via the pulmonary um, arteries all the way. The blood can go from the right ventricle to the lungs very easily because now they have a sudden decrease in pulmonary vascular resistance. And this happens because when the baby takes the first approach to crying, when the baby cries for the first time, that gasp of air is extremely important for the decreasing of pulmonary vascular resistance. So that is why when a baby is born, are we worried or are we not worried that the baby is crying or not crying? If the baby doesn't cry after birth, are we worried? Yes or no? Yes, we are very worried if the baby doesn't cry after birth, because if the baby doesn't cry, then the baby cannot breathe. The baby still has the lungs filled with fluid. So when the baby cries, okay, you will see that a lot of physicians, I'm not sure if they do it over here, but a lot of physicians back home, they will hang the baby upside down and slap him on the butt a couple of times, okay, so that the baby can start crying. So have you guys seen that? Have you guys ever seen a physician do that? Okay, hang the baby upside down, slap him very gently on the butt twice so the baby starts crying. Right. So they do that. That's because the baby can have have the first gasp of air, which is needed, have the first gasp of air, which is needed to decrease the pulmonary vascular resistance. And once that decreases, all the blood from the right ventricles can flow very directly to the lungs via the pulmonary artery. So that's that. Okay. So that's basically all the things that you basically need to know. Another high yield piece of information is the development of the alveoli. Basically, at the alveolar stage, you have at birth, you have 20 to 70 million alveoli at birth. And by the time at 8 years, that is the alveolar stage, it stays from the 36th week to 8 years. At 8 years, you have 300 to 400 million alveoli. So as a matter of fact, right now, people like me and you, how many alveoli do you think that we guys that we have on an average, adults? Okay, close to around 300 to 400. That's basically close to around 300 to 400. That's how much alveoli we have. Okay. Okay. So that's all about the lecture on stages of lung development. That's all about the lectures on the stages of lung development. Okay. Okay. So that's that. Okay. Okay. All right. So can we move on to the next part of the stage? Okay. Can we move on to the next one? Yes. Okay. Very good. Okay. So we wanted to talk about two other things before we move on to the next one. The two other things that we wanted to talk about are some lung malformations, congenital lung malformations. Congenital lung malformations. Okay. Before we jump into this, let's see the congenital lung malformation. So we're done with this stage over here. Okay. Okay. So before we move on, what are the questions that you guys will receive? First of all, you guys need to know the names of each and every stage for the purpose of doing well. Then you guys need to know the weeks, especially this week over here. Okay. Pseudoglandular stage. This week is important because at this week, if the child is born, then the child cannot breathe. So this is important over here. This stage is also important because from this one, the child is viable for life. So from the canonical state, the weeks of these ones are important. The pathology of the first one is important. Okay. This is a high yield question. Okay. Then over here, the pneumocytes developing at 20 weeks. This is high yield. Respiration is capable at 25 weeks. This is high yield. Okay, how many alveoli does the child have at birth? This is very high yield. And this is very high yield. That is pulmonary vascular resistance. Okay, this is a 3-star. Okay. Okay. Okay, so why do I only mark the high yield ones? Does anyone have any idea? Why do I only mark the high yield ones? Okay. Do you guys see a lot of posts in Usmle Step 1 preparation forum or any other group where they talk about how they can finish first aid in 2 weeks and 3 weeks? Yes or no? Okay. Do you guys think that they read each and every line when they finish first aid in 2 weeks or 3 weeks? No. Well, what they do is they read the high yield ones. So these are the high yield ones which you need to focus on when you are reading the first aid for the second time. Okay, so that's that. That's why I'm okay. Another one is, if you guys do not even want to look at this one, if you guys can look at this diagram over here, then this can help you. But to be honest, I always found this a little bit confusing. Okay, so I always stuck to the text. I never really stuck to the diagram because I personally find it a bit confusing. So that's that. Okay. So now what we're going to do is we're going to move on to congenital lung malformations. Okay, congenital lung malformations, we have two problems over here. First of all, we have pulmonary hypoplasia and we have bronchogenic cysts. Okay, pulmonary hypoplasia is basically a poorly developed bronchial tree with an abnormal histology. It is associated with congenital diaphragmatic hernia and bilateral renal agenesis. So basically, what is pulmonary hypoplasia? As I just said, that the young baby that they breathe in and they expulse, I mean, they aspire and cause expulsion of the amniotic fluid. So if that does not happen, will there be proper expansion of the lungs? Yes or no? Will there be proper expansions of the lungs? Yes or no? Fast answers, guys. If the young baby, if they do not aspire and expulse the amniotic fluid, can lungs expand? Okay, what's happening over here? What's happening is this is the young baby's lungs. Okay. Okay, this is the young baby's lungs. So all this fluid that we have, that is the amniotic fluid, when the baby breathes in the amniotic fluid, okay, the amniotic fluid goes over here. Okay. The amniotic fluid goes to the lungs, and this causes lung expansion. Okay, this causes lung expansion. So if the amniotic fluid is not there, so this will not cause lung expansion. So this will result in the formation of a disease known as pulmonary hypoplasia. Pulmonary hypoplasia, if it's there, the reasons are basically two. First of all, we have congenital diaphragmatic hernia. That is an association. Okay, this is not a cause, this is an association that pulmonary hypoplasia can be associated with congenital diaphragmatic hernia on the left side. Okay, because why? Because on the right side, you have the liver. On the left side, there is an empty space. And if the lungs are underdeveloped over here, then there's a high chance for the abdominal structures, especially the stomach, to herniate all the way up over here. Okay, there's a high possibility for the stomach to herniate all the way up over here. So there could be an association of congenital diaphragmatic hernia. So that's that, especially on the left side. So left-sided congenital diaphragmatic hernia. Another one is Potter syndrome. Potter syndrome is happening due to bilateral renal agenesis or autosomal recessive polycystic kidney disease to some extent. That what we have is we have decreased urination of the baby, and the amniotic fluid is decreased. So you will have oligohydroamnias. Less amount of fluid is available for lung expansion, and that's why you have pulmonary hypoplasia. Okay, and another one is bilateral renal agenesis. Bilateral renal agenesis. Okay, that is pulmonary hypoplasia. Okay, next one that I want to talk about is bronchogenic cysts. Bronchogenic cysts, they're basically what they are is, if we have the normal development of the lungs from the budding of the foregut. That is the ventral foregut that they usually bud and they form the respiratory diverticulum, right? If that is not normal, okay, if there's an abnormal budding of the foregut, then what we have is we have the dilatations of the terminal bronchioles. Okay, we have the BC stands for bronchogenic cysts. Okay, what is the pathology? The pathology is basically that over here, when we talked about the little baby, okay, we talked about that this is the, okay, right? This was the respiratory diverticulum. From the distal end of the respiratory diverticulum, we had the formation of the lung bud, okay? And due to the proper budding of the lung bud, we had the formation of the bronchial buds, main stem bronchi, segmental bronchi, and then the tertiary bronchi, right? So that's the normal budding. If there's abnormal budding, okay, if there is abnormal budding of the lung bud, then what you can get is the dilatation, you have dilatation of terminal bronchioles. Terminal bronchioles. Basically, if we can think about the fact that, let's say that all this budding that was happening, okay, and then you have another budding from over here, okay. So these things that they have to happen in a sequence, that is the sequence that we studied over here. The sequence was this sequence, that is lung bud, trachea, bronchial buds, mainstem bronchi. It has to happen in this sequence. If there is abnormal budding, meaning that let's say one sequence happens before another sequence, that's that there's underdevelopment of one of the buds, then we can get what we say is that this terminal bronchiole over here, they cannot fully function and perform and form the next sequence in the formation, that is the respiratory bronchiole. So what you can get is you can get dilatations of the terminal bronchioles. Terminal bronchioles. So this dilatations of the terminal bronchioles, this results in the formation of a cyst known as bronchial cyst. This bronchial cyst, if we can see an X-ray of the bronchioles of the bronchogenic cyst, okay, if we can see an X-ray of the bronchogenic cyst, they appear, okay, okay, 1 second. Okay, this is a good one. Okay, this is a bronchogenic cyst. Okay, bronchogenic cyst, what they appear is they appear as a round, sharp, well demarcated, and somewhat of fluid-filled density. They see how over here this is not dark. I mean black. Okay, black means that there is presence of air over here. That since there is no air exchange or gaseous exchange, this part is completely or more or less white over here. So this is a fluid-filled density of, okay. Generally what happens is they are not that high yield in the sense that they can remain asymptomatic, okay, but there are multiple questions which you will get just on this X-ray alone, okay. So next time you guys receive an X-ray like this with these type of sharply well demarcated things over here in a young child, then try to consider bronchogenic cyst. The bronchogenic cyst can cause the pathology is that they can cause recurrent respiratory infection. Recurrent respiratory infection. So that's that. Are we clear about this? Are we clear about this? Yes or no? Bronchogenic cysts. Are we clear about bronchogenic cysts? Okay. Repeat hyperplasia. Which hyperplasia? Hyperplasia. Dr. Prabhasi, would you be kind enough to ask the question again, please? Pulmonary hypoplasia, not hyperplasia. Pulmonary hypoplasia. Hypoplasia. Hypoplasia basically means that the lungs have not developed due to decreased amount of amniotic fluid that is present for inspiration. If there is no amniotic fluid, then there is no expansion of the lung. then the lungs cannot expand and the lungs remain small and hypoplastic. This is known as pulmonary hypoplasia. If you have pulmonary hypoplasia, the cause can be bilateral renal agenesis, in which you have decreased amniotic fluid available. And another one is there can be an association of congenital diaphragmatic hernia, because if the lungs are small, there is a potential space over here. This allows for creation of a vacuum through which the stomach, I mean the abdominal contents, can herniate to the thorax. This is known as congenital diaphragmatic hernia. Okay, are we clear? Bronchogenic cyst in particular causes lower recurrent respiratory infection. They can cause recurrent respiratory infection. Mostly it depends on where they are. If they are presented in the lower part of the lungs, then over there they can cause infection. If they're there in the upper part of the lungs, they can cause um, the upper part of the lung infection. So basically, the location of the infection is not that high yield, but the fact that the knowledge is required for the fact that it causes recurrent respiratory infection, that is more important. Are we clear? Are we clear about this? So that we can move forward, guys. Okay. Okay. Next one. Next one that we want to go to. The next one that we want to go to is this one. Okay. Club cells. What are club cells? Club cells are basically, these are non-ciliated, low columnar cuboidal, columnar to cuboidal cells with secretory granules. Okay, they are located in the bronchioles and they degrade toxins, secrete a component of surfactant, and they act as reserve cells. Okay, so basically club cells, if we have to, club cells are really important. Okay, club cells, let's talk a little bit about club cells for 2 minutes because you will receive a lot of questions from club cells. Okay, let's talk about the functions of the club cells. Club cells have, okay, so club cells. What are the functions of club cells? First of all, the functions of the club cells are degrade toxin, degrade toxin. So the toxins which we are inhaling, or that's there in the blood to some extent, can be degraded by the club cells. Okay, then what they do is secrete component of surfactant. Three functions basically. Okay, and reserve cells. Reserve cells meaning that they act as reserve cells, meaning that they can be used meaning that they can be used to fill up the lung cells if by any chance there is degradation of the normal lung cells. So that's that. So they act as reserve cells. But the highest yield ones are these two, that is toxin degradation and secretion of a component of surfactant. And once again, they're non-ciliated, columnar to cuboidal. The locations are usually, they are located in the bronchioles. They are located in the bronchioles. Okay, so club cells, that's that. Okay, next one. The next cells that are important, the next cells that are important that we want to talk about are alveolar cells. Okay, out of the alveolar cells, now we have the development. Now we are going to talk about that cell, that is the pneumocytes cells. And before we talk about pneumocytes, what is the week of development of the pneumocytes? At which week does the pneumocyte develop? At which stage does the pneumocyte develop? At which stage? Okay, canalicular stage, very good. So at the canalicular stage, the pneumocytes develop. Okay, so now what we have is we have the type 1 and type 2 pneumocytes. Okay, so what we need to understand that, okay, that type 1 and type 2, Okay. A lot of students, they are not sure about type 1 and type 2, which one is more high yield, and asked more questions about. Okay. Type 1 and type 2. Okay. Type 1 is less superior. Type 2 is more superior, meaning that type 2 are stronger cells. Okay. Type 1 is okay. Type 1 is not that strong, meaning that type 1 are squamous cells. Squamous. Type 2 cells are type 2 cells are cuboidal cells. Cuboidal cells. Cuboidal cells. Okay. My apologies. 1 second. Type 2 cells, cuboidal cells. Okay. Now, the next one is, what are the functions of type 1 cells? The function of type 1 is that the type 1 are only used for alveolar lining. Alveolar lining of the alveoli. And okay, and the type 2 cells, okay, type 2 cells have very distinctive function. Okay, this is very important. I can't even stretch how many questions you guys will receive from this information only. That is, they act as stem cells. Okay, before I talk about this, okay, can anyone tell me what is the stem cell of the GI system? What is the stem cell of the GI system? Guys, come on, please. I'm really counting on you guys. What is the stem cell of the GI system? GI, gastrointestinal system. Stem cells of intestine. Why are we not more confident? What are the stem cells of the GI system? Okay, where are the stem cells of the GI system located at? Are Have you guys heard of Paneth cells? Paneth cells? Do you guys think Paneth cells are the stem cells of the GI system? Okay, so what is the stem cell of the GI system? Okay, what are the stem cells of the liver? Liver stem cells. Stem cells of the liver. Thank you. Okay. Stellate cells or okay. Okay. Homework for you guys. Okay. Homework for you guys. Stem cells of the body. Okay. Or as a matter of fact, this is not fair. I'm gonna do this homework for you. Stem cells of the body. All the stem cells of the body. I will write it down. Okay, but I really thought you guys would remember the stem cells of the GI system. So I will mention them. I will mention all the stem cells of the body. Don't worry. But for the lungs, the stem cells are the type 2 pneumocytes. Okay, please do not forget. You can forget about the stem cells of any part of the body, no problem. But you cannot forget that the stem cells of the lungs are type 2 pneumocytes. What do we mean by stem cells? Stem cells mean that these cells, they have the potential to develop into any part of the lungs if there is lung damage. If there is lung damage, the type 2 pneumocytes can proliferate and differentiate into any type of cells. Okay, so this is known as stem cells. Another function of the lung of the type 2 pneumocytes are S for stem cells and S for surfactant. Surfactant. Type 2 pneumocytes are responsible for the secretion of lung surfactant, from especially from over here. Okay, especially from over here. Okay, Uh, as you can see that this part, this one over here, and this is the type, this is the organelle in type 2 pneumocyte. This is the type 2 pneumocyte as a whole. This organelle is called lamellar bodies. Lamellar bodies. Okay, from the lamellar bodies, they secrete the surfactant. This information of lamellar bodies is high yield for emboss. Okay, if anyone is solving emboss, this is important. If anyone is solving UWorld, you guys will not be asked about lamellar bodies, but you guys will be asked about the this one over here. Okay, so are we sure about type 1, type 2 pneumocytes, their function? Yes? Yes or no? Yes or no, guys? Okay, very good. Another cell that we want to talk about, another cell that we want to talk about are alveolar macrophage. Macrophage. Alveolar macrophages, they are basically that they phagocyte. They are phagocytes that is they phagocytose foreign materials, right? Phagocytose foreign materials. What happens is, let's say that this is the lungs, right? Let's say that this is the lungs, and you inhale bad air. For example, in smoking, example when we smoke, okay, we have a lot of foreign particles that is coming inside the lungs, and this alveolar macrophages, they get activated, and then they cause phagocytosis. Okay, they cause phagocytosis of those foreign materials. Okay, they cause phagocytosis of those foreign materials, and when they phagocytose those foreign materials, they release a very important substance. This substance is known as alveolar proteases. Alveolar proteases, what they do is that they degrade this substance into little breakdown pieces, right? They degrade this substance and then they completely consume the substance. So all this inflammatory process, what this does is this causes the release of a very important inflammatory substance that is known as cytokines. So you have the release of cytokines from here. Okay, so that's the mechanism of that's the mechanism of action of alveolar macrophage. Are we clear about the mechanism? First of all, dirty air, phagocytosis, release of alveolar proteases, and then the release of cytokines. These are the step-by-step mechanism of the alveolar macrophages. Another one is if we have, for example, if we have another one that is, okay, if this is the heart, right? And then over here, what we have is we have the lungs, right? Okay. If the left heart, left ventricle, if it does not function properly, there will be buildup of the blood. This will go in the lungs. In the lungs, this RBCs will be broken down. The red blood cells will be broken down by the macrophages in the sense that the RBCs will be engulfed. Okay, the RBCs will be engulfed. And then once again, there will be release of alveolar proteases and breakdown of the RBCs. And when RBCs are broken down, you have the release of hemosiderin. And these hemosiderins are, they will get deposited over here in the macrophages, okay? These are known as hemosiderin-laden macrophages. I'm pretty sure every one of you are well acquainted with hemosiderin-laden macrophages or heart failure cells. These are known as heart failure cells, okay? And they are found in the setting of a heart failure or pulmonary edema. In the setting of a heart failure, we get pulmonary edema, either that or in the case of an alveolar hemorrhage, for example, in a traumatic lung injury. So that's that. Okay. Are we clear about the three cells of the lungs? Are we clear about the three cells of the lungs? Yes or no? Okay. So what are the two cells that are responsible for the formation of the lung surfactant? What are the two cells that are responsible for the formation of lung surfactant? Type 2 and club cells. Okay. Write it down. Write it down over here. Surfactant. We'll write it down. Surfactant. Cells responsible are type 2 and club cells. Write it down like this so that every time, so that the next time when you guys revise first aid, this is the first thing that you can see when you read this page, because this is the only thing that was extremely high yield. Okay, good. Next point is I want to talk a little bit about lung surfactant. That is, what is lung surfactant? What is lung surfactant? Lung surfactant basically is the secretion that is created from type 2 pneumocytes and club cells, which will decrease the alveolar surface tension, right? This will decrease the alveolar surface tension. And if there is decrease of alveolar surface tension, meaning that let's say that this is the alveoli, right? So the surface tension of the alveoli, um, meaning that the alveoli, they always have a tendency of collapsing. Okay, the alveoli, they always have a tendency of collapsing. And what happens is the lung surfactant, they form. Okay, they are present over here like this. The lung surfactant, they're present over here like this. So what they do is they decrease the surface tension. They prevent the alveolar collapse. So now the alveolar, they have a harder probability of collapsing. And at the same time, what they do is they decrease lung recoil. What do we mean by decrease lung recoil and increase compliance? Increased compliance means that when air comes, okay, 1 second. Increased compliance mean that when air comes right now through here, the lungs, they have increased ability to stretch. So the ability of the lung to stretch properly is known as compliance. And this happens because the lungs are not collapsed because of the presence of the surfactant over here. So they increase lung compliance. So that's why surfactants are really important. Okay. When we talk about lung surfactant, there are two components of the lung surfactant which we need to talk about. There are two components of the lung surfactant that we need to talk about. The components are basically, okay, 1 second. Okay, so the lung components are basically, they're composed of two things. That is the surfactant over here. Okay, they are basically composed of two things that is very high yield. Number one is, and 1 second, 1 second. I don't want to confuse something over here. Okay, right. Basically, what this is they are composed of lecithin, and what they have is sphingomyelin. I wasn't sure about the I wasn't sure about the spelling. I'm sorry. Okay, sphingomyelin. I don't want to confuse the spellings. Okay, my apologies. Lung surfactant. Basically, they have basically what we have is they have lecithin and sphingomyelin. But over here, what happens is the lecithin and sphingomyelin. Okay, in the lecithin, okay, they are mainly composed of lecithin. Out of lecithin and sphingomyelin, you have mainly lecithin. From this lecithin, there's another high yield thing that is dipole methyl phosphatidyl choline. In short, what we say is DPCC. DPPC. My apologies. DPPC. Okay. So this lecithin, that is the most important part of the lung surfactant. Okay, that is the most important part of the lung surfactant. Um, this is basically composed of another. This usually contains another type of a substance that is dimethyl phosphatidyl choline. Okay, and this synthesis of the lung surfactant, they usually begin at 20 weeks. That makes more sense, obviously, because at 20 weeks you have the formation of the type 2 pneumocytes, and when the type 2 pneumocytes, they are formed, they cause the secretion of the lung surfactant, and the completion of the lung surfactant are completed at 35 weeks of gestation. At 35 weeks of gestation. So basically, if we have a young baby who is born at any time before 35 weeks of gestation, Okay, before 35 weeks of gestation, then the lung surfactants are not enough. So what we do is we treat the baby with dexamethasone or corticosteroids, basically corticosteroids, because the corticosteroids, what they do is they stimulate the type 2 pneumocytes to secrete more large lung surfactant. And at the same time, this can actually save the baby from neonatal respiratory distress syndrome. Okay, so that's that. Okay, are we clear about this? Are we clear about this? Okay. There's another high yield question which you will receive. Is we for lung, that is a lung maturity screening test. For lung maturity screening test, we have this another high yield question, that is for lung maturity you have to have this one thing, that is when we talked about surfactant we talked about two things, lecithin, lecithin and sphingomyelin. What we always have to understand that the lecithin has to be more than the sphingomyelin. So both of them are usually there in the surfactant, but the lecithin has to be more than the sphingomyelin because the lecithin contains diethyl methylphosphatidylcholine. And so what we do is, in a clinical setting, we do a ratio of the lecithin to sphingomyelin in the amniotic fluid. That is before the baby is born. Okay, that is before the baby is born, because we want to check for fetal lung maturity. Fetal lung maturity. For fetal lung maturity, we do a test that is known as LS ratio test. Okay, what is LS ratio test? LS ratio basically means lecithin to sphingomyelin ratio test. Okay, over here, let's say that we find lecithin. Let's say that the amount of lecithin that we find is 8. Okay, this is just an imaginary number. Say we find 8, and sphingomyelin we find 2. Okay, so the ratio is basically 8 by 2, so that is 4. Okay, 4. Anything above 2, anything above 2 is healthy. Okay, anything less than 1.5 is not healthy, meaning that these babies have high possibility that they have surfactant deficiency or decreased formation of lung surfactant, and they have high chance of development of neonatal respiratory distress syndrome. Okay, so if we want to draw if you want to draw a graph, okay, if we want to draw a graph, basically what we should expect in a normal baby is that the lecithin should increase and the sphingomyelin should decrease. Okay, this is sphingomyelin, this is lecithin for proper lung surfactant components. Okay, and the lecithin to sphingomyelin ratio, the lecithin to sphingomyelin ratio should increase. Okay, L by S ratio. L by S ratio should be increased, meaning that it should always be more than 2, and it keeps on increasing throughout, because the more lecithin increases, the more sphingomyelin should decrease. So it should keep on increasing like this. But if for some reason, if the graph doesn't happen like this, then there's a possibility for these children to have neonatal respiratory distress syndrome. So do not forget, you guys will get a graph like this in your U world. Okay, in your U world, you will get a graph like this where they will have this one and this one for lung surfactant components, and they will ask you which one is lecithin, which one is sphingomyelin. And I'm pretty sure you guys know by now, A is lecithin, B is sphingomyelin. You guys will receive this question in U world, very high yield. Okay, very high yield. You guys will receive this question in U world, and always remember that more than 2 is responsible for proper lung development. Okay, what happens if we have what happens if we have um, surfactant deficiency? Guys, what happens if we have surfactant deficiency? Now, are we all aware that if the surfactant is deficient, then there could be alveolar collapse? Okay, you guys just said it, alveolar collapse. If there is alveolar collapse and we do an X-ray of a patient, okay, if we do an X-ray of a patient, okay, okay, over here, okay, 1 second. Okay, basically what I wanted to show you guys was that this picture looks like a glass, meaning that the normal baby should have lung development. Normally, you should see the air fills in the lungs, right? But these babies, they do not have any gaseous exchange, more or less, because all the alveoli are collapsed over here. So there's absolutely no dark setting. You can see a very mild shadow of the heart, like this over here, this over here. This shows that the heart is actually dilated. Okay, this means that the heart is actually, they have to work more to push the blood against the high vascular resistance. So that's the reason why the heart is a bit dilated. But over here, as a whole, which you can see is that there's a ground glass appearance of the lungs. This is known as neonatal respiratory distress syndrome. If you do an X-ray, you see that there is a ground glass appearance on the lungs. Okay, so ground glass appearance of the lung. So that's that. So this is what you can see in surfactant deficiency or neonatal respiratory distress syndrome. What are the causes of surfactant deficiency or neonatal respiratory distress syndrome? There are some risk factors, not causes. Basically, what we have some risk factors. The risk factors of neonatal respiratory distress is, number one, we have prematurity, which is completely understandable, meaning that if the baby is premature, we said that the surfactant has to be developed at 35 weeks, at which it's mature, right? It's matured at 35 weeks. If it's like anything less than 35 weeks to 30 weeks, then we can have the neonatal respiratory distress syndrome. Okay, so that's that. Another one is um, another one is previous C-section. Okay, what's happening over here? Why do we have babies with C-section? Why do we have decreased lung surfactant? Okay, when the baby, okay, C-section, is C-section a natural way of delivery, or is this a modern way of delivery? C-section, is this a natural way of delivery? This is a modern way of delivery. Okay, thousands and millions of years ago, no one would know of C-sections where they can cut open the uterus, okay, and then take the baby out. But according to nature, okay, what nature allows during vaginal delivery, normal vaginal delivery, what nature does is, okay, this is actually interesting. Okay, what nature does is, okay, nature loves you, but the love is tough love. What does that mean? That means that basically, when the baby is born during the normal vaginal delivery, the first part of the life of this young baby, the baby has to suffer and the baby has to stress a little bit. Meaning, what's going on over here? When the baby is coming out through the normal procedures of the birth, that is through the normal vaginal delivery, the process for the baby is actually, it's tough than normal baby. So the baby has to fight to come out. Basically, the baby has to fight to come out, and the baby has to uh, basically basically the baby has to survive. So the baby has to go for fight or flight. The baby has to fight, right, or flight. So baby, or obviously, since the baby can't flight, the baby will fight. The baby will fight to come out. What happens when the baby fights to come out to the normal vaginal delivery? The baby has increased amount of stress, right? So the first thing in life that the baby experiences is stress, and this stress secretes a high amount of glucocorticoids. Glucocorticoids, right, from the baby's adrenal cortex. So, for example, cortisol. This cortisol helps the lungs. Okay, this cortisol helps the lungs establish the proper surfactant. This cortisol helps the lung establish the proper surfactant needed to prevent the neonatal respiratory distress syndrome. So this stress is actually good. This is actually good for the baby. Okay, but as time passes by, things are much more easier for everyone, including babies. So now we lead a very, what can I say? We lead a very easy life. Okay, so easy love over here. So this baby over here, he was sleeping in the mother's womb, and then very gently, the physician, the gynecologist, cut open the mother's uterus, and the baby got out. The baby did not even experience any stress. So, do you think that these babies, will they have more glucocorticoids or less glucocorticoids than the babies of normal vaginal delivery? Yes or no? Less glucocorticoids. So, if they have less glucocorticoids than these babies of cesarean section, they have a high chance of developing neonatal respiratory distress syndrome. Are we clear about this? This whole story about tough love and this and that is just so that you guys do not forget how normal vaginal delivery is needed for your surfactant production. Okay, so that's that. Okay, next one. Okay, next one is after the baby is born with the surfactant deficiency, what we do is we try to give the baby oxygen. Do we not? Yes or no? Do we try to give the baby oxygen? No. What this oxygen can do, if the oxygen is not given and if it's not given in proper amount, this oxygen, this therapeutic oxygen that we give the baby with neonatal distress syndrome, can actually cause more harm than good. Okay, more harm than good. What can this oxygen cause? This causes, okay, this causes three things. Um, R I B. RIB stands for retinopathy, I for intraventricular hemorrhage, and B for bronchopulmonary dysplasia, RIB. These are the three things that can happen when you give the baby therapeutic oxygen to treat neonatal respiratory distress syndrome, because you have to understand that these babies, they're already having a difficult time breathing air because they do not have enough lung surfactant. On top of that, if you keep on giving excess oxygen more than it's needed, then this oxygen can cause more harm than good by creating free radicals, or in some other way they will cause these three damages: retinopathy, intraventricular hemorrhage, and bronchopulmonary dysplasia. Okay, are we clear about this? Yes, are we clear about this? Okay, repeat please. Lecithin with the gestation before 36, 35 weeks. Basically what's happening is you have the development of the lung surfactant at 20th week of gestation, because then the type 2 pneumocytes are being formed. Lung surfactant usually contains two components: lecithin and sphingomyelin. But sphingomyelin has to decrease and lecithin has to increase, because lecithin is the most important component of the lung surfactant. The lecithin contains diapalmethyl phosphatidylcholine, and the ratio of lecithin to sphingomyelin has to be more than 2. So before 35 weeks of gestation, what we do is, before 35 weeks of gestation, if we want to make sure that the mother, I mean the baby, has enough surfactant or not to survive after birth, we can do a lecithin to sphingomyelin ratio. If the lecithin to sphingomyelin ratio, if it comes to more than 2, then this is a good sign. If it's anything less than 1.5, then it's not a very good sign. Are we clear about this? Okay. Okay. Next question. Is there an explanation to why there's retinopathy of premature or rib happening? Okay. So first of all, the first disease is retinopathy of prematurity. In retinopathy of prematurity, what happens is when we give supplemental oxygen to these babies who do not have proper lung development, these supplemental oxygens, what they can do is they can cause, first of all, they can cause free radical damage. That's number one. Number two, if they do not cause free radical damage, in retinopathy of prematurity, you have disorganized growth of the retinal blood vessels. Okay, so there is angiogenesis. There is angiogenesis angiogenesis of the retinal blood vessels that causes retinal scarring, fibrosis, and retinal detachment. Okay, that causes retinal scarring and retinal detachment. That is why you get retinopathy of prematurity. Are we clear about this? Are we clear about this? Yes or no? Okay, so what's happening in retinopathy of prematurity? In retinopathy of prematurity, we have the therapeutic supplemental oxygen, which are causing angiogenesis of the retinal blood vessels, disorganized growth, scarring, and fibrosis. So that's why you can also get retinal detachment to some extent, and that's why these patients get damages. Okay, so that's exactly what I mean. Any more question? Okay. Any more question? No. Okay. So that's that. Okay. Okay. So we are done with respiratory embryology. Okay. We are done with respiratory embryology. So before we begin respiratory anatomy, do you guys want to take a break or do you guys want to begin respiratory anatomy right away? Laplace's law. Okay. Laplace's law is important. We will talk about Laplace's law in the physiology. Okay. We'll talk about the Laplace's law in physiology. Okay. Okay. So what about the break or no break? Do we take a break now or do we take a break a little bit later? The law, yes, Laplace's law. Laplace's law, we will talk about it in physiology. Okay. Okay, how long do you guys want to take the break for? Okay, 10 minutes. Okay. We will take the break for 10 minutes, not more than that. And we will come back at 11:28. Okay, is everyone back from their break? Okay. Okay, very well. We are going to begin with the respiratory anatomy. So that's what we're going to do. Before we jump into the text of the respiratory anatomy, okay, I want to start with a diagram. 1 second. Okay. So I want to start with the diagram of the respiratory anatomy. Basically, I want to divide the respiratory tract into two zones, that is the conducting zone and the respiratory zone. And I want to talk about the linings of each and every zone. So conducting zone and respiratory zone. The first one that I want to talk about is conducting zone. Okay. Another one that I want to talk about is the respiratory zone. So before I talk about the respiratory zone, let's start with the conducting zone first. So the conducting zone of the lung basically means that it's air conduction. There is no gaseous exchange, no gaseous exchange of the lung. Okay. So the parts of the lungs that is responsible for air conduction and no gaseous exchange are, at first we have the trachea. Okay. Then we have the trachea. Then we have the bronchi, right? We have the bronchi over here. Okay. Then we have the bronchioles. Okay, we have the bronchi, then we have the bronchioles. Okay. Then we have the terminal bronchioles. Okay. So basically we have T, B, B, T. Okay, do not forget this. T, B, B, T. Okay. So over here, the reason why you need to remember which one is the conducting zone, which one is the respiratory zone, is because there's a severe change in the linings of the respiratory tract from all the way from over here. So from the trachea to the bronchi, the lining is pseudo pseudo stratified ciliated columnar epithelium with cartilage. Cartilage. So if we have to draw the cartilage, that would be like this. The lung, they have cartilages over here with cartilages. Okay, so pseudo stratified ciliated columnar epithelium with cartilage. So the pseudo stratified ciliated columnar epithelium with cartilage. There's also another thing with cartilage and goblet cells. So we have the mucus producing cells over here. So the reason why we have pseudo stratified ciliated columnar epithelium and goblet cells and the cartilages is that the goblet cells, they secrete the mucus. So whenever we breathe in anything dirty, they get stuck in the mucus, and then by the beating of the cilia back and forth, we have the mucus going all the way up, right? And then we either cough it out, or we eat, or it goes from the trachea all the way up to the esophagus, and we swallow it. That's the normal physiology. So that's how we remove the debris from over here. Okay. Once we enter the bronchioles, okay, once we enter the bronchioles, we have simple ciliated columnar epithelium. It's not pseudo-stratified, it's simple ciliated columnar epithelium. And over here, we have with club cells. Okay, so the bronchioles, they like to go to the club. Okay, so bronchial club. So simple ciliated columnar epithelium with club cells. Okay, so that's that. Then we have the last one, that is terminal bronchial. Terminal bronchial, we have it's the same thing, simple ciliated columnar epithelium, simple ciliated columnar epithelium to simple cuboidal epithelium. So what happens is there is a transformation. So over here you have columnar, columnar, and then you have cuboidal, cuboidal. So this transformation. Okay. And you also have the club cells. So they also take the terminal bronchioles to the club with them. Okay, so club cells. So that's that. So that's the conducting zone of the lung. Are we all clear about the conducting zone of the lung? Are we all clear about the conducting zone? Yes? Okay, good. Next one. Next one is the respiratory zone. Okay, next one is the respiratory zone. The respiratory zone of the lungs, we have basically two parts of the respiratory zone. Two parts of the respiratory zone. First of all, we have the respiratory bronchioles, and we have these balloons over here. Okay, these balloons are basically alveolar sacs. So respiratory bronchioles and alveolar sacs. Respiratory bronchioles, the lining of the respiratory bronchioles are, since over here you have the exchange to cuboidal, so it's very simple to understand that this is simple cuboidal epithelium. Okay. And this cuboidal epithelium will transform to the transformation into simple squamous epithelium and basically to the alveoli, which contains a squamous. So over here, what we have is, if we draw a transection, okay, over here, if we draw a transection, then these are the squamous epithelium of the alveoli. Okay, this is the type 1 pneumocyte. If you guys remember correctly, type 1 pneumocytes are squamous. We talked about this before. And then over here, you have another cell. Okay, what are these cells? These cells are type 2 pneumocytes. That are the stem cells and surfactant secreting cells, right? And you have another cell over here, which we talked about in the lung. That is the alveolar macrophage cells, alveolar macrophage cells. So pseudostratified to simple columnar to cuboidal to squamous. Okay, so that's that. Are we all clear about this? Are we all clear about this? Yes or no? Yes, okay. So I will give you guys exactly 2 minutes to read this because this is extremely high yield. Okay, this is really, really high yield. You guys will receive a lot of questions from this in respiratory physiology. Okay, if needed, there's a picture like this in first aid. That is this picture. Okay, I need you guys to take a picture of this in your phone or your laptop if needed for the days that we're going to study the respiratory system. Make this picture your screensaver for the laptop or for your phone. So every time you open your phone, you see this picture, because this picture will have a lot of questions. Okay, we'll have a lot of questions from over here. So I'll give you guys exactly 2 minutes. In 2 minutes, just learn about what's going on over here and these things. And I will ask you guys the question. Okay. Another high yield thing, which I think I should mention, although it's common, it's common knowledge and it's a very basic knowledge. That is, I want to talk about the smooth muscles. Basically, you have smooth muscles in all the layers, right? Except in the alveolar sacs. For obvious reasons, the alveolar sacs, they do not have any smooth muscles. So that's that. Okay. Smooth muscles are responsible for the constrictions and dilatations of the bronchioles, right? This is where the drugs, where, for example, the beta 2 agonist, they come and they work over here for dilatation. So that's that. So smooth muscles are basically there in all the layers except the alveolar sac, which is basic knowledge. Okay, so I'm going to come back over here and keep this, learn about the conducting and the respiratory zone for 2 minutes, and then I will come and ask you guys questions. Okay, so 2 minutes on the clock. It's 11:41. I'll give you guys time till 11:43. Okay. Okay, are we done? Okay, good. So what is the epithelial lining of the trachea and the bronchi, please? Can I get some fast answers, please? Epithelial lining of trachea and the bronchi. With cartilage or without cartilage? Okay. What is the epithelial lining of the bronchioles and bronchioles? Okay, what is the epithelial lining of the terminal bronchioles? Terminal bronchioles. Okay. What is the epithelial lining of the respiratory bronchioles? Okay. What is the epithelial lining of the alveolar sac? Okay. What is the segment of the conducting zone where there could be bronchiectasis? Bronchiectasis. Fast answers, please. What is bronchiectasis? What is bronchiectasis? Anyone? Okay, no problem. If you guys cannot, nope. Dilatations and secretions, irreversible. Okay, no problem. You know what? I will. Okay, we will master this concept in 1 second. Before we will master this concept. I just wanted to see if we were sure about what bronchiectasis is. I won't discuss this right now. I just wanted to see if you guys knew, but no problem. We will discuss this in 1 second. Okay. Now go back to our text over here. Let's go back to our text. In our text, we have the respiratory tree, which consists of the conducting zone and the respiratory zone. What are the questions you will receive from this text over here? First of all, the large airways, they consist of nose, pharynx, larynx, trachea, and bronchi. Airway resistance is highest in the large to medium-sized bronchi. Underline this. Airway resistance is highest in large to medium-sized bronchi. Small airways consist of bronchioles that further divide into terminal bronchioles, which has the least airway resistance, Okay, so that's that. Okay, so the conducting zone, they warms, humidifies, and filters air, but they do not participate in gaseous exchange. So this is known as the anatomical dead space over here. So this is exactly what I said, that over here you do not have any space for gas exchange to occur, that is oxygen to enter and carbon dioxide to leave. So this is known as the anatomical dead space. Through this one is known as anatomical dead space, basically meaning that there's no gaseous exchange over here. Okay, so over here you have the cartilage and the goblet cells, which will extend to the end of the bronchi. And then they talked about the epithelial lining, that is the pseudostratified ciliated epithelium, which makes up the epithelium of the bronchus and extends to the beginning of the terminal bronchioles, which we had just said. And then it transitions to cuboidal cells, which we have already mentioned. So we have said most of the things. And then we already talked about mucus and how they are responsible to be escalated. That is the mucus secreted by the goblet cells. They are beaded by the cilia left and right all the way up to the lungs. And then either they're coughed out, sneezed out, or they are swallowed in the esophagus. So that's that. And then the airway smooth muscles extend to the end of the terminal bronchioles. And we also talked about this. So that's that. Next one is respiratory zone, lung parenchyma. Okay, if you have to receive one question from over here, it's this one, airway resistance. It's highest from the large to the medium-sized bronchi. This is high yield. Okay, if you have to receive question, you might receive question from this sentence by itself. Respiratory zone is basically the lung parenchyma. This consists of respiratory bronchioles, alveolar ducts, and alveoli. They participate in gas exchange. Mostly they are the cuboidal cells, then simple squamous epithelium up to the alveoli, and then the cilia, they terminate in the respiratory bronchioles. Okay, so we basically, the cilia are up to here. Okay, so we know that. Always remember that the cilia are present absolutely in the conducting zone. Why? Because the conducting zone has the goblet cells. They are conducting air. So when when the lungs are conducting air, they want to make sure that the air they're conducting is fresh air, right, which is devoid of any debris or foreign particles. So the mucus that is being produced over here, these mucuses, the they will trap the debris and the foreign particles, and then by the beating of the mucociliary, by the cilia, by the beating of the cilia, this mucus will go all the way up. Once again, same information. I'm repeating this over and over again so you guys understand the importance of this information. That the conducting zone will be the only layer which contains the cilia. That's that. Okay, so that's that. So they will terminate in the respiratory bronchioles, meaning that from the respiratory bronchioles, you do not need to conduct any more air. You need to have the gaseous exchange. So the cilia are not necessary in the respiratory bronchioles. And then if by any chance there is still more debris that's coming here, right? For example, the debris which could not be removed by the mucus, if there's more debris which comes here, there's still nothing to worry about, because over here we have our friend, that is alveolar macrophage. They will clear out the excess debris. Okay. Are we clear about this? Okay. Next question. We have a question which is, Dr. Alveolar sac is squamous and cuboidal, because one type is squamous and type 2 is cuboidal. It's mostly squamous, meaning that the lining is squamous by type 1. Okay. And then you have the type 2 in between. That's that. Okay. Okay. Are we clear about this? Are we clear about this? Okay. Okay. Next one. Can we move on to the next page? Okay. Lung anatomy. We are done with the respiratory tree. Now we are moving to the lung anatomy. Okay, the lung anatomy, the lung has basically the right lung has 3 lobes and the left has
two lobes. So left has less lobe. Left means less, right means more, more means three. Okay, so try to remember it like this. That left lung has three lobes. In have we all seen an actual human lung in our anatomy classes in medical university? Have we all seen cadaveric lung? Cadaver. Yes. Okay. So it should not come as a surprise to all of us that the right lung has three lobes and left lung has two lobes, because I'm pretty sure we all know that. Am I correct? Yes. Yes or no? Are we all accustomed to that knowledge? Okay. So right lung has three, left lung has two, right? Left lung has three lobes, right? The left lung has three lobes? No? Okay, good. Left lung has two lobes. Right lung has three lobes. Okay, do not mix this up. I'm going to tell you why, because you will receive a lot of questions from this in USMLE, where they will try to have sneaky questions. Okay, where they will try to test your knowledge on this. Okay, so the lobes, do not mix this up. Okay, okay. So we have to learn about the relation of the pulmonary artery. Um, and the right lung and the left lung. Okay, so the relation of the pulmonary artery with, okay, I just want to show you a picture. Okay. Mhm. Okay, so this is the hilum of the lung. Okay, this is the hilum of the lung. Hilum means this is the part of the lungs through which all the segments that needs to enter the lung, they enter. We have the bronchus, artery, pulmonary vein. Okay, so the relation of the pulmonary artery to the bronchus is over here. You can see that the pulmonary artery, the right-sided pulmonary artery is in front. The right-sided pulmonary artery is in front of the bronchus. Over here, if we see the left lung, we see that the uh, over here, if we see that the uh, left lung, in the left lung, the pulmonary artery is superior to the bronchus. In the left lung, the pulmonary artery is superior to the bronchus. So the very common mnemonic which we use, a common mnemonic which we use for the relation of the pulmonary artery to the bronchus is RALS. RALS stands for, what is the mnemonic? The mnemonic is RALS. RALS means right pulmonary artery is anterior to the bronchus in the right hilum, and left pulmonary artery is superior to the bronchus in the left hilum. Okay, clear? Are we clear about this? Okay, okay, okay. Repeat, please, of course. So, do we know what the hilum of the lung is? Hilum? Do you know what the hilum of the lung is? Okay. We are studying the relation of the pulmonary artery to the bronchus. Okay, we are studying the relation of the pulmonary artery to the bronchus. That's what we have to understand for doing well in the exam, and also for surgical purposes as future surgeons. Okay, especially for cardiothoracic surgeons. Over here, if you see a hilum, how will you know that this is the right hilum or the right or the left hilum? Basically, in the right hilum, the right pulmonary artery will be anterior to the bronchus. For example, this one, right hilum, the pulmonary artery is anterior to the bronchus. In the left hilum, the pulmonary artery is superior to the bronchus. Okay, that's all I mean. That's all I said. RAL. This is the mnemonic. Are we clear about this? Yes or no? Okay. 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 Next one. Next one is another high yield piece of information, which is okay, which is very important. That is, if we draw a lung like this. I mean a trachea, and then the trachea divides into right and left bronchus. Okay, the next one is higher, right bronchus, and this is the left bronchus. The right-sided bronchus is the right-sided bronchus is actually the right-sided bronchus. This is the right-sided bronchus is actually more wider, more vertical. Okay, it's wide, it's vertical, and it's actually shorter. Okay, and then the left one is actually more slimmer and less vertical, meaning it's more horizontal. So if we had, for example, this is the esophagus. Okay, if we had a food substance, okay, if we had a food, for example, this entered your lungs, or a foreign body which entered your lungs, which one of these bronchus has the highest chance of receiving the food particle or the foreign substance, the right side or the left side? Okay, the right side. Why? Because the right side is more vertical and the right side is more wider and the right side is shorter. Okay, now we have to understand it depends on the right side of the lung. First of all, the right side of the lung, first of all, the right side has three lobes. Okay, the right side of the lung, they have three lobes, right? Now, when we are lying down, okay, when we are lying, okay, first of all, let's draw the lobes of the lung. Okay, first of all, let's draw the lobes of the lung. Okay, this, okay, okay. Upper lobe, middle lobe, and the lower lobe. Okay. If we are lying down, if we are supine, if the food enters, the food will go and lodge in the superior segment of the right lower lobe. The food will come and lodge in the superior segment of the right lower lobe, that is over here. That is if you are supine. Okay, so supine, supine, meaning that you are lying down. When we are lying down, for example, if any foreign particle they go, then the food will go into this part. Because if you think of yourself lying down and this patient over here is lying down over here, then this is the segment. The lower part comes in close contact with this one over here. So this is a higher chance for, due to gravity, the food to come all the way and get lodged into the upper part of the right lower lobe. Okay, if we are lying on our right side, if we are lying on our right side, then the food will come and enter into the right upper lobe. Okay, so this is for lying on right side. If we are on the right side of, let's say we are, like how we have a lot of people who sleeps on one side. For example, I personally, like I'm also a sided person, meaning I cannot sleep supine. So a lot of people, they sleep on the right side or on the left side. So if anyone is lying on their right side, this portion of the lung, this portion comes in close contact over here. Then due to gravity, if any foreign particles were to aspirate inside the lungs, it will go to the right upper lobe. Okay, right upper lobe when they're lying on the right side. And if you are standing, okay, if the patient is standing, then the gravity is all the way over here. Then the gravity is all the way over here. And due to that, this gravity, when upright, the food will come and lodge all the way over here. So right lower lobe. So this is when upright. Okay. I cannot even stretch how important this knowledge is, this piece of knowledge is, because you will receive so many questions just on the location and the position of the patient. Once again, if the patient is lying on the right side, the right upper lobe is close to the trachea, so that due to gravity, this will come and lodge in the right upper lobe. when the patient is lying on the right side. If the patient is lying supine, okay, all the way supine, then the food will lodge in the right upper lobe of the right upper part of the lower lobe, okay, right superior segment of the lower lobe. And if the patient is standing or upright, then it will lodge in the right lower segment of the right lower lobe. Okay, that's that. Are we all clear? Are we all clear about this? Did you guys draw this diagram? Did you guys draw this diagram? Yes or no? Okay. Once again, I'll give you guys another time. 1 minute, just 1 minute. Read, look at this diagram. I'll ask you guys questions. Okay. Okay. So if a person is standing upright, where will the food get lodged in? Can I get some fast answers, please? If a person is upright, where will the food get lodged in? Right lower superior segment or inferior segment? Inferior segment. If a person is lying supine, where will the food get lodged in? Right lower lobe. In right lower lobe, superior segment. Very good. If a person is lying on the right side of the bed and food aspirates, where will it get lodged in? Right upper lobe. Okay. Okay, good. Okay. So are we all clear and confident about this? Yes. Yes. Okay, good. Okay. Next one. The next one that I want to talk to you guys about, the next one that I want to talk to you guys about is the diaphragmatic structures. Okay. So the structures of the diaphragm. Structures of the diaphragm. Okay. Do we have any student over here who loves eating eggs? Who loves eating eggs? Anyone? Eggs? I know there are a lot of people who are huge fans of eggs. I personally am a huge fan of eggs, except the yolk. I can stand the smell. Okay. The white portion is high in protein, so the yolk is something which I really don't like. But do we have any? Yes. Okay. How many eggs do you guys have on a daily basis? 3 to 5. Okay. Do you know how many eggs I have every day on a daily basis? Okay. No, I never talked about this. Okay. 10. Very good. Dr. Karbasi is so smart. Okay. 10 eggs. I have 10 eggs. And do you know I have a perfect time at every day. I eat those 10 eggs exactly at 12 p.m. because that's the optimal time for my protein intake. In Okay, I have 10 eggs exactly at 12. Exactly at 12 p.m. every day when I take that short break with you guys. Okay, I go down and I eat 10 eggs. Okay, so tomorrow I'm going to ask you how many eggs I eat and what's the time. So you guys will not forget that I eat 10 eggs at exactly 12. Okay, so um, okay, you guys thought that we were discussing about something, but we really weren't. What I was discussing about was I ate 10. 1 second. I ate 10 eggs at 12. I ate 10 eggs at 12. Okay, so I usually eat 10 eggs. How many eggs do I eat? 10, especially the white part. And I eat them at exactly at 12 p.m. Okay, because it's the perfect time for my protein intake. So this mnemonic of me eating 10 eggs at 12, okay, this is the mnemonic which is needed for the three perforations of the diaphragm. Okay, basically at T8, meaning at thoracic 8, the diaphragm is perforated by I, that is inferior vena cava. At T10, that is at thoracic level 10, the diaphragm is perforated by esophagus. Esophagus. At T12, the diaphragm is perforated by aorta. Are we clear about this? Easy, very, very easy. Next time you think about diaphragm, think about my egg eating habits. Okay, Dr. Heidey eats 10 eggs at 12. So I ate 10 eggs at 12, meaning inferior vena cava at T8, esophagus at T10, okay, and aorta at T12. Okay. Okay, no, when eating eggs, think about this. Okay, think about this whenever you want. I have no problem. Okay, but just do not forget this. Okay, when you eat eggs, when you don't eat eggs, okay, when you do whatever you want to do, no problem. When you receive a diaphragmatic question, that is the actual time when you need to think about this. So do whatever you have to do to remember this mnemonic. This will help you. Okay, so at T8, you have inferior vena cava, but with inferior vena cava, you have a nerve. Okay, the nerve is right phrenic nerve. Right phrenic nerve. With the esophagus, you have the vagus. Vagus and vagus together. Esophagus and vagus. Okay, and with this one, you have, as a matter of fact, you have no nerves at T12. What you have is you have azygous vein and you have thoracic duct. Okay, easy. Is this easy? Yes or no? Good. Okay, so fast answers, please. At T8, which structure perforates the diaphragm? Fast answers. At T, which structure diaphragm? Okay. At T10, which structure perforates the diaphragm? At T12, which structure perforates the diaphragm? Okay. Okay. All right. Okay, very good. Which structure, along with the inferior vena cava, perforates the diaphragm at T8? Okay. Which structure with esophagus perforates diaphragm at T10? Structure with the aorta perforates the diaphragm at T12? Structures, azygous vein and thoracic. Very good. Okay. With that said, we're done with respiratory anatomy. Are we clear about this? Are we clear about respiratory anatomy? Okay. Okay. Jump into. Let's take a very quick jump into the respiratory physiology. Okay. Respiratory physiology, very high yield. Okay. Respiratory physiology is very high yield in the sense that you will have a lot of questions from the respiratory physiology. And to some extent, I kind of find it a bit fun to read respiratory physiology. Who else finds respiratory physiology fun? Okay. Who else finds respiratory physiology fun to read? Let me see. Okay. Okay, good. If you do not find fun in respiratory physiology, hopefully you might find some. Okay, but how about this lecture today? Are you guys understanding the lecture, not me group? Okay. Are you guys understanding the lecture? I'm taking things very slow, nice and easy at a very slow pace so that we understand. The reason why I usually like to take things nice, slow at the beginning. Didn't you guys realize that I have this habit of taking things very slow at the beginning and then at the end I take it extremely, I just kick it up one pace up, then we finish it. Okay, you know what the reason is? The reason is if you understand the basics really easily, like in reproductive system. Do we have all the students here today who has done reproductive system with us? Yes. Yeah. In the reproductive system, did you guys realize that in that at the beginning we took things very slow, nice and easy. Everyone tried to make sure everyone understood everything, and then at the end we kicked it up one piece and we finished everything at one go. Okay, that's because if you guys have a strong basic right now, then near the end during the pathology and the pharmacology, you guys will not need a lot of understanding to do because you guys will have the perfected the basic. Okay, so having that in mind, let's move on to respiratory physiology. Okay, this is actually high yield. What we're going to do is we're going to learn about the lung volumes. Lung volumes. Okay. What is the difference between volume and capacity? Fast answers, please. What is the difference between volume and capacity? There's exactly only one difference, guys. Capacity is multiple volume. That is correct. Meaning that more than one volume is known as lung capacity. More than one volume is known as lung capacity. So lung volumes basically, how many types of lung volumes do we have? How many types of lung volume do we have? First of all, we have, okay, one second. First of all, we have inspiratory reserve volume. Okay, inspiratory reserve volume. What is inspiratory reserve volume? This means that the air that we can still breathe in after a normal inspiration. Let's say that we inspire normally. Okay, let's say that right now um, we are taking a we are breathing normally over here, right? And then all of a sudden, right now, I ask you to take your maximum inspiration all the way, right? Like that. So you take the maximum inspiration. Okay. So first of all, in order to understand that, we have to understand tidal volume. What is tidal volume? Tidal volume is the air that moves into the lung, outside and inside with each and every quiet inspiration. Air that moves into the lung with each quiet expiration. So right now we are, okay, we are breathing in, breathing out, breathing in, and then breathing out again. And then all of a sudden now I ask you guys to take your maximum inspiration, like, okay. And then this is your inspiratory reserve volume. Okay. So, okay, my apologies for acting out the inspiration, but I'm not sure if you guys heard. I tried to inspire. I'm not sure if you guys did the same or not, but if you guys did, then this is your normal inspiration. And then all of a sudden you take the maximum amount of air that you can inspire after a normal expiration. This is your inspiratory reserve volume. Okay. After that, we have another air volume, that is expiratory reserve volume. Expiratory reserve volume is basically air that can be breathed out after a normal expiration. Okay, so let's say we take that normal breath and then we're releasing that, this one. Okay, and now, okay, once again you breathe normally and then you expire normally. Okay, and then you expire some more, like this one. Okay, so you expire some more, air out. That is your air that can be breathed out after normal expiration. So that's that. So that is expiratory reserve volume, air that can be breathed out after normal expiration, air that can be breathed in after normal expiration, and air that is breathed in and out at normal expiration. So these are the three volumes. We have another volume, the last volume, that is the residual volume. Residual volume is the air in the lungs after a maximum expiration, meaning that you can try to expire as much as possible. But even after you expire as much as possible, there will always be some amount of air that is left behind, which you cannot expire. If you were to expire this air out, the lungs will collapse. So it's absolutely impossible for you to expire this amount of air out. So that's that. Okay. So this is basically the lung volumes. Are we all clear about this? Yes or no? Are we all clear about this? Yes or no? Okay. Okay. So that's that. Okay. Which one? Reserve, re- which volume? Expiratory reserve volume. Expiratory reserve volume is the maximum amount of air that can be breathed out after a normal expiration. This is the maximum amount of air you can breathe out after a normal expiration. Okay, are we clear? Okay, 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 good. Next one. Now we are going to talk about the capacities. Inspiratory capacity, okay? Inspiratory capacity. What is inspiratory capacity? Capacity, once again, basically means the addition of two volumes. So basically, inspiratory capacity is inspiratory reserve volume plus tidal volume, meaning that in inspiratory capacity is the air that can be breathed in after a normal exhalation. So basically after this normal exhalation, this is when we are exhaling, right? You're exhaling now normally. And when I ask you to take a deep breath in, you take a deep breath from here to up to here. This is the normal amount of air that you can breathe in after a normal exhalation. So inspiratory capacity is your capacity to breathe in air after a normal expiration. So inspiratory capacity is inspiratory reserve volume plus. Okay, do we have any question? Is the voice breaking? Is the voice breaking? Can you guys hear my voice, or is the voice breaking? Okay. Okay. Okay, good. Okay, so inspiratory reserve volume is inspiratory capacity is inspiratory reserve volume plus tidal volume over here. Okay, then next one is functional residual capacity. Functional residual capacity is functional residual capacity is the volume of the air which stays after normal expiration. This is the volume of air which stays after normal expiration. So if I ask you about the volume of air which stays after normal expiration, the volume of air which stays after normal expiration, then which are the two volumes that should be added up for functional residual volume? Fast answers, please. Functional residual capacity, not volume. It's okay, the normal amount of air that stays in the lungs after a normal exhalation. So this is the normal capacity of the lung after a normal exhalation. Am I correct? So the functional residual capacity is basically expiratory reserve volume plus reserve volume. Expiratory residual volume plus My apologies, expiratory reserve volume plus residual volume. Okay, that's that. So these are the two capacities. Okay, are we all clear about inspiratory capacity and functional residual capacity? Yes. Okay. Next one is vital capacity. Vital capacity is the maximum volume of air that can be expired after a maximum inspiration. Okay. Basically, if we take a deep breath, okay, so when you blow out your candles, when everyone is done singing happy birthday for you, right? And you want to make sure that, let's say that you are 20 years old, so you have 20 candles on the cake, and you want to blow out the 20 candles. What do? You take a deep breath, and then you blow it out as hard as possible. Am I correct? Yes or no? Do you do that? Yes or no? During your birthday, when you want to blow out all the candles on the cake, do you take a deep breath in and then try to blow out as hard as possible? Okay. Okay, so that is basically what you are doing is you are using your vital capacity. Vital capacity is you are taking a deep breath in. So this is the maximum amount of air that can be expired after a maximum inspiration. So from over here to all the way over here to all the way over here. This is your vital capacity. So vital capacity is basically inspiratory reserve volume plus tidal volume plus expiratory reserve volume. Okay, are we clear? Are we clear about this? Yes or no? Okay, next one. Next one is total lung capacity. This is one of the most easiest things to understand. Total lung capacity is basically this one plus this one plus this one plus this one. Total lung capacity is all the way from here to all the way here. That is total lung capacity. Okay. And the total lung capacity is usually 6 liters. Okay. 6 liters. Okay. Are we clear about this? Questions asked. Questions will be asked based on, I will show you examples of the questions that will be asked. It will be asked in a bit of a complex way. They will ask you questions regarding the changes that happens to the volumes in different types of lung pathologies. So that's how the questions will be asked. Okay, they will not ask you what are the volumes in respiratory, I mean in functional residual capacity, this and that. Okay, so in order to understand the questions, you have to understand this volumes first. Are we clear about the lung volumes? Okay, are we going too slow or is the pace of the lecture okay? This should be just understood. Yes, this should be just understood, and then later we can apply this knowledge. Okay. 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 All right. Next one. Okay. And then, do you guys remember that we talked about the anatomical dead space? Anatomical dead space. Yes. Remember the anatomical dead space? Okay. Which part of the lungs are the anatomical dead space? Conducting zone. Okay. If, for example, let's say a person has pneumonia, will the dead space of the lung increase or decrease? Dead space of the lungs will increase because gas exchange will not happen. Okay. Now, have you guys heard physiological dead space? Yes. Physiological dead space is basically the dead space. Okay. What is physiological dead space? Physiological dead space is anatomical dead space plus alveolar dead space. Alveolar dead space. In a normal healthy individual, alveolar dead space, should there be any alveolar dead space where gas exchange is not happening in a normal healthy patient? We say that in a normal healthy patient, the alveolar dead space is 0. Can we say that? Yes. Okay. So if a healthy young patient, the physiological dead space is anatomical dead space plus 0. So basically, physiological dead space is equal to anatomical dead space. Are we clear about this? Yes? Yes or no? Are we clear about this? Okay. Okay. There's also uh, an equation for physiological dead space. Physiological dead space, the equation is basically, okay, one second. The equation for physiological dead space is tidal volume, one second, physiological dead space, VD. VD means physiological dead space, is equal to tidal volume, means VT, into into PaCO2. PaCO2 means arterial partial pressure of carbon dioxide minus partial pressure of carbon dioxide in expired air divided by partial pressure of carbon dioxide. This is the equation. This is the equation. How does this equation derive? That is a very long lecture. Okay, that is a very long lecture. But we just want to find out how much of the expired carbon dioxide has left. So we subtract this from the normal partial pressure of carbon dioxide. Then we find the ratio of this one to this one, and then we get this one. We multiply it by this one, and then we get the physiological death rate. How the equation comes into the derivative, that is a very different type of lecture. Okay, so this is that. And you guys do not need to memorize or keep in mind the equations as of right now, because you guys have a whole page for equations in first aid at the back, which I told you guys. Do you guys remember what I told you guys about the equations for the back? That for the first 15 minutes of the USMLE step one, instead of looking at the tutorial, you guys should use that 15 minutes to write the equations on a piece of paper. Okay, so that's all. That's your time for understanding. Right now, just read through the equations. So that's that. Okay. Are we clear about physiological dead space? Are we clear about physiological dead space? Physiological dead space should be equal to anatomical dead space. If physiological dead space is not equal to anatomical dead space, then there's a problem. Especially there is a dead space in the alveoli for which gaseous exchange is not happening. That's that. Then the next one is physiological dead space has an equation that is tidal volume multiplied by partial pressure of carbon dioxide subtracted by expired partial pressure of carbon dioxide divided by partial pressure of carbon dioxide again. So that's that. Okay. Okay, the next thing that I want to talk about is ventilation. Okay, the next thing that I want to talk about is ventilation. In ventilation, we have two types of ventilation, that is minute ventilation and alveolar ventilation. Ventilation. Okay, what do you mean by ventilation? Ventilation means the amount of gas that enters the lungs per minute. Yes or no? The total amount of gas that enters the lungs per minute. Yes or no? Is that the definition for ventilation? Right? So what is ventilation? Ventilation is the total amount of gas that enters the lungs per minute. So Basically, if we breathe per minute, the normal respiratory rate is, for example, 16 to 18 breaths per minute, right? So what is the normal amount of air that is entering and exiting the lung? So basically, we have minute ventilation, that is tidal volume into respiratory rate, into tidal volume. That's that. And what is alveolar ventilation? Alveolar ventilation is gaseous exchange that is happening per minute. So alveolar ventilation is tidal volume minus dead space volume into respiratory rate. Very easy to understand. Okay, these are the two equations. Okay, are we clear about this? Are we clear about this? Okay, easy or to understand? VD means physiological dead space. Okay. 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 Next one. Okay. How can we be asked to calculate? Yes, you will be asked to calculate, but we will talk about this at the end of respiratory system, because then we will read the equations. So when we read the equations, you guys will get 15 minutes at the starting of USMLE Step 1 exam, where you guys, instead of doing the tutorials, you guys should use that time to write the equations on a piece of paper, and then you guys can fill those in with the numbers and do the calculations. Okay. Okay. Can we move forward? Okay. Can we move forward? Guys, is everyone okay? Can we move forward with the lecture? Okay. Okay, next one. Next one that I want to go to. Okay, so we're done with all of this. Okay. Okay. The next one that I want to go to is basically lung and chest wall. Okay, so lung and chest wall, meaning that you have the lung and chest wall, you have three terms: elastic recoil, compliance, and hysteresis. So we have to understand what we mean by these three terms. So first of all, elastic recoil is basically this is the tendency of the lungs to collapse inward and chest wall to spring out. So okay, so you have two forces that is working like this, and the chest wall that is like this. Okay, so these two forces working in the opposite direction, this is then known as the elastic recoil. This is the tendency to collapse and the chest wall to spring out. So this is known as the elastic recoil. At functional residual capacity, the airway and lung and the airway and alveolar pressure equals atmospheric pressure, and intrapleural pressure is negative. The inward pull of the lung is balanced by the outward pull of the chest wall, meaning that at functional residual capacity, what we have is this tendency to collapse is cancelled by this force over here. So this force and this force is canceling each other out. So as a result, the lung stays at a normal level, at a normal elastic forceful level. So the system pressure is atmospheric. Pulmonary vascular resistance is at a minimum over here. So over here, the pulmonary vascular resistance is at a minimum. Are we clear about this elastic recoil? This is basically the inward force canceling the outward force. Okay. You guys only have to understand the terms. No questions will be asked regarding this as of right now, except their association with the pathologies. Are we clear? Okay. Next one. Next one is compliance. Compliance means the ability of the lungs to stretch. This basically means change in lung volume for a change in pressure. So V by P. So V, lung compliance, is basically V by P. Change in volume by change in pressure. When the pressure of the lung changes, the volume of the lung also changes. This is inversely proportional. That is, the compliance is proportional to wall stiffness, and it is increased by surfactant, meaning that if the walls are stiff, the compliance, if there's increased wall stiffness, meaning that the lungs have less ability to stretch, so increased stiffness means decreased compliance, vice versa. If there is decreased stiffness, increase lung compliance. Lung compliance, once again, is the ability of the lungs to stretch out. So that's that. Okay, so what are the causes of increased compliance? Compliance mean when the lungs can easily fill with air. That is emphysema and aging. What happens in emphysema? Well, okay. Instead of talking about emphysema right now, we will talk about this in 1 second. But just remember that in emphysema, what we have is the lungs are easily stretched out because what we have is we have dilatations of the alveoli, right? We have the breakdown of the term of the septas and the alveolar septors, and then the alveoli can easily stretch out. So that's what happens in emphysema, and the compliance of the lungs increases. And when the compliance of the lung decreases, that's when the stiffness of the lungs increases. And the causes of lung stiffness increasing is fibrosis, pneumonia, and ARDS. Fibrosis, pneumonia, and ARDS. So this is what's happening. Have you understood compliance and elastic recall? Have you understood compliance and elastic recall? Okay. Compliance and elastic recall. Elastic recall is basically the inward force cancelled by the outward force. And as a result, the lungs are staying at a normal pressure. The both of the forces are canceling each other out. This is known as the elastic recall. Compliance basically means the ability of the lung to stretch, and in cases, for example, stiffness of the lung. For example, if you have pulmonary fibrosis, the lungs cannot stretch, so the compliance decreases and stiffness increases. And in cases where the lungs can stretch more easily, for example, during emphysema, you have the destructions of the alveolar septa. So the lungs can stretch more easily because the stiffness of the lungs have decreased. So that is known as compliance, increased compliance and decreased compliance. Okay, are we clear about this? Are we clear about this? Yes. Increased stiffness will decrease compliance. Okay. Okay. Next one. Next one is this graph over here. As you can see that in this graph, this is transpulmonary static pressure. And this is the volume. What's happening, if you can look at the graph, is that you have the volume of the lung. So this is the chest wall compliance. As the chest wall compliance increases, the lung compliance also increases. Okay, and this is that. So basically, this means that okay, if this is the lungs and if this is the chest wall, right? Okay, 1 second, please. 1 second. Okay, can you guys hear my voice? Is the voice clear? Okay, okay, all right. Okay, so we were finished with elastic recoil and compliance. Can we move on to hysteresis? Okay. So the thing is, now we're going to study about hysteresis. Okay, hysteresis, hysteresis over here. Okay, okay. So we were talking about this one before I had to receive that phone call. Okay, my apologies. So basically, this basically means that you have the lungs and you have the chest wall. So the more the expansion of the chest wall, the more expansion of the lungs. So that's the vice versa. So chest wall compliance is directly proportional to lung compliance. As simple as that. There's nothing more, nothing less. Then we have hysteresis. Hysteresis is basically lung inflation. They follow a different pressure volume curve than lung deflation due to need to overcome surface tension forces in inflation. Okay, so in surface tension forces, okay, it's easier for the lungs to deflate than the lungs to inflate. Why? Because the lung surfactant over here, which is lining the lung like this, the lung surfactant, which is lining the lungs like this, it's harder for the air to push the lungs out than then for the lungs to collapse. So in order for the lungs to inspire, they have to overcome this force, the surface tension force over here, in order to expire like this. I mean, in order to inspire like this, and then when they inspire, the surfactant will pull the lung inwards, and the air can leave the lungs again. Okay, so if you try breathing right now, okay, if you try breathing, when you take a deep breath in, you guys will realize that it's easier to let the air out than it is for the air to come inside the lung. So can you guys try it for me one more, just only one time? Try to take a deep breath in. See that it's easier, that the process of expiration is easier than the process of inspiration, because in inspiration you have to let the air in, the air has to overcome the force of the surface tension, and the surface tension force will let into the will result in the expansion of the lungs, and with the expansion of the lungs, the surfactant will now push the air out, so it will be easier for the lungs to expire. So this is lung hysteresis. Are we clear? Are we clear about lung hysteresis? Easy to inspire. I mean, harder to inspire, easy to expire. Okay. When you inspire the lungs, they have a surfactant, okay, which is providing a force to the lungs. So when you inspire, the force of the air has to overcome the surfactant's force. And when they overcome, okay, let me draw another small diagram. Okay, let me okay, okay, 1 second. Okay. Um, okay. Guys, is it possible to end the lecture a bit early today, like right now? Okay, because the network is being extremely horrible, and we can add an extra hour tomorrow. Is that okay? Because the net keeps on disappearing for the bad weather but that we have today, and we apologize for this. So instead of continuing this forwards, let's start the lecture a little bit early tomorrow, and if needed, we can start like at 8:30 or right 8:30 ish, and we can finish at 2, so that way we can have a proper um, discussion and stay ahead of the schedule. Is that okay? Is that okay, guys? Yes. Okay. Does everyone agree? I'm really sorry. My apologies, but to be honest, I feel like yes, we can start tomorrow at the same time. No, we will start tomorrow a bit early. We will start tomorrow a bit early at 8:30. Okay, so that we can finish a bit earlier. Okay, so let's finish the lecture over here for today. It's a bit complete, and we will start the lecture. An hour earlier tomorrow. Okay. Yes, 8:30 tomorrow. Okay. Okay, all right. Okay, perfect. Thank you so much for understanding. And I'll see you guys tomorrow at 8:30 a.m. Okay, if you guys have any question, please send me an email and I will respond back to you. Okay, all right. Thank you so much.